All right, welcome to Rad Onc Smart Review, and get ready because today we're really plunging into the world of cell signaling, specifically, how it all connects to radiation biology and cancer.
Yeah, and it's fundamental stuff.
Totally. Now, why should you care? Well, understanding these signals, how cells talk to each other and themselves, it's basically the bedrock for figuring out how radiation therapy actually works, how it hits the cancer cells and, hopefully spares the normal tissue.
Exactly. And we'll be using a really helpful framework to guide us through this, the hallmarks of cancer.
Ah, yes, the hallmarks. Essential.
Absolutely. It doesn't matter if you're a PGY2 just starting out or, prepping for boards, getting a solid grip on cell signaling is
Yeah.
just, well, it's non-negotiable. It explains the why behind so much of what we do. So yeah, let's dive into this framework, the hallmarks of cancer.
Right. So back in 2000, Hanahan and Weinberg published this landmark paper. It took the super complex reality of cancer and kind of boiled it down to 6 core abilities a normal cell needs to acquire to really go rogue.
It's like the rule book for becoming malignant. Yeah.
Exactly.
And what's so powerful about it is how it distills this incredibly messy process into, well, manageable concepts. Once you grasp these core ideas, you start seeing the common threads across different types of cancer, different treatments. So where should we start? Maybe the first hallmark.
Sounds good. Let's do sustaining proliferative signaling.
Okay.
So imagine a car, right?
Yeah.
And the gas pedal is just jammed down permanently. That's sort of what's happening here. Normal cells, they only grow and divide when they get the right signal.
Hmm.
But cancer cells,
Huh.
they figure out how to keep those go signals firing constantly.
Yeah, they hotwire the system.
Totally. Often involves things like overactive oncogenes, like RAISE, that's a key signaling switch.
Yeah.
Or maybe just way too many growth factor receptors on the surface, like EGFR. They just keep telling the cell, grow, grow, grow.
And, just like a car needs brakes, normal cells need ways to stop growing. Which brings us straight to the second hallmark, evading growth suppressors.
Right, the stop signal.
Exactly. Tumor suppressor genes, I think RB1, TP53, those are the crucial breaks. Cancer cells find ways to cut the lines, basically, often through mutations that just knock these genes out. So the stop signals might still be there, but the cell just isn't listening anymore.
Okay, so the accelerator's stuck, the brakes are gone. What's next?
Well, then there's avoiding self-destruction. Resisting cell death, that's the third one.
Apoptosis. Programmed cell death.
Precisely. Normally, if a cell gets damaged or it's just not needed, it undergoes this orderly self-destruct sequence. But cancer cells, they become survival experts. They might overproduce proteins that block apoptosis, like BCL2, or they might mess with the proteins that carry out the execution, like P53 or BAX. Essentially, they become incredibly hard to kill.
Okay, so they keep growing, ignore stop signs, and refuse to die. What else?
The fourth one is really fascinating, enabling replicative immortality. This is where cancer cells basically achieve biological immortality. They refuse to get old and stop dividing.
The Hayflick limit thing, right?
Exactly. Normal cells can only divide a certain number of times, but cancer cells often flip a switch, reactivating an enzyme called telomerase. And reactivating telomerase isn't just about dividing forever. It's like they achieve this escape from normal cellular aging. It lets them keep dividing and piling up more mutations. We definitely need to circle back and talk more about telomeres and telomerase later.
Definitely. Okay, number 5.
Number 5 is inducing angiogenesis, building a blood supply.
Right? Gotta feed the beast.
Exactly. Like a growing city needs roads, a growing tumor needs blood vessels for oxygen, nutrients. Cancer cells actually send out signals, VEGF is a big one, telling nearby blood vessels to sprout new branches into the tumor. Without this, tumors really can't grow beyond a couple of millimeters.
And the last of the original 6.
Activating invasion and metastasis. This is what makes cancer so dangerous, its ability to spread.
Yeah, metastasis is the real killer often.
It is. Cancer cells learn how to break away from the primary tumor, invade nearby tissues, hop into the bloodstream or lymphatics, travel, and then set up shop somewhere else, like little colonies.
Wow. Okay, so those are the original 6. And it's key, right, that a cell usually needs several of these, not just one.
Oh, absolutely. It's typically a multi-step process. Acquiring just one usually isn't enough for full-blown cancer.
And the story didn't end there, did it? There were updates.
Correct. In 2011, Hanahan and Weinberg revisited the framework. They added 2 emerging hallmarks and 4 enabling characteristics, adding more layers to the picture.
Okay, what are the emerging ones?
First is deregulating cellular energetics or metabolism. Cancer cells often rewire how they produce energy to fuel all that rapid growth.
The Warburg effect.
That's the classic example, yeah, where they favor glycolysis, this less efficient way of making energy, even when there's plenty of oxygen around. It seems weird, but it gives them the building blocks they need for making new cells quickly.
Okay. And the second emerging hallmark?
Avoiding immune destruction. Our immune system is usually pretty good at spotting and eliminating abnormal cells. But cancer cells develop these sneaky tricks to either hide from immune cells or even suppress the immune response, like an invisibility cloak.
Clever and deadly. Okay, and the enabling characteristics, what are those?
So these aren't hallmarks per se, but they're underlying conditions that make it easier for cells to acquire the hallmarks. First is genome instability and mutation.
Ah, so things are just more likely to go wrong in the DNA.
Exactly. Cancer cells often have faulty DNA repair systems. This leads to more mutations, more chromosome abnormalities, which accelerates the acquisition of those hallmark capabilities. And interestingly, this is a key vulnerability that radiation exploits.
Right, by causing more DNA damage. What's the second enabling characteristic?
Tumor-promoting inflammation. Sometimes chronic inflammation around a tumor can actually help it grow.
Wait, inflammation helps the tumor. That seems counterintuitive.
It does, right.
Yeah.
But that inflammatory environment can supply growth factors, survival factors, help with angiogenesis, even help cells invade. It's complex, a bit paradoxical.
Okay, now I know there was an even more recent update, like 2022, mentioning things like EMT epigenetics.
That's right. Things like phenotypic plasticity via EMT, non-mutational epigenetic reprogramming, the role of microbiomes, senescent cells. The field keeps evolving. But honestly, for our purposes today, and definitely for board exams, focusing on the 2000 and 2011 versions, those original 6, the 2 emerging, and the key enabling ones, that's the core knowledge you need.
Got it. So original 6 down cold, plus be aware of the key 2011 additions.
Precisely. That's high-yield stuff.
Okay, you mentioned earlier we should dive deeper into one of these. Let's do enabling replicative immortality, telomeres and telomerase. This stuff is fascinating.
It really is elegant biology. So,
Yeah.
most of our normal cells are somatic cells. They can't divide forever. They have this built-in limit, the Hayflick limit we talked about.
Right. They divide maybe 50, 70 times and stop.
Yeah, they enter this state called replicative senescence. It's an irreversible growth arrest. And the molecular clock counting down those divisions, that's the shortening of telomeres.
Okay, so tell us about telomeres. What are they exactly?
Think of them like the little plastic tips on the ends of your shoelaces, aglets.
Aglets. Right. Stop them fraying.
Exactly. Telomeres are protective caps at the ends of our chromosomes. They're made of repetitive DNA sequences in humans. It's TTG, repeated over and over, plus some proteins. They stop the chromosome ends from looking like broken DNA, getting tangled or fusing together.
Okay, protective caps. So why do they shorten?
Ah, that's the end replication problem. It's a bit technical, but basically the machinery that copies our DNA polymerase, it can't fully replicate the very, very end of a linear DNA strand, particularly something called the lagging strand.
So a little bit gets lost each time the cell divides.
Exactly, like those aglets getting worn down a tiny bit each time you tie your shoes. Over many divisions, the telomeres get progressively shorter. That's the mitotic clock.
And when they get too short.
The cell senses it as damage. Critically short telomeres trigger a DNA damage response. This usually activates pathways involving P53 and PRB, leading to that replicative senescence. The cell stops dividing, enters G1 arrest permanently.
Okay, and you can sometimes see that with a marker, right?
Yeah.
SA beta-gal?
Yes, senescence-associated beta-galactosidase. Its activity often goes up in senescent cells, so it's used as a marker in research.
Now, cancer cells obviously don't like stopping.
Yeah.
How do they get around this?
Well, if a cell has already managed to mess up its P53 or RB pathways, maybe evading growth suppressors, Hallmark 2, it might bypass senescence and just keep dividing even with short telomeres.
That sounds dangerous.
It is. Those unprotected chromosome ends can fuse together, break apart, rearrange, leads to massive genomic instability. This phase is called crisis, and most cells actually die here via apoptosis.
But some survive. The ones that become cancerous.
Some survive. And the main way they do this, how they achieve that replicative immortality in about 85 to 90% of cancers, is by reactivating an enzyme called telomerase.
Okay, telomerase. The key enzyme. What does it do?
Telomerase is fascinating. It's a ribonucleoprotein part RNA, part protein. The RNA part, PERK, has the template for the T-tag repeat. The protein part, TERT, is a reverse transcriptase.
Reverse transcriptase, like HIV, uses RNA to make DNA.
Exactly like that mechanism. TERT uses the TORC RNA template to add those T-tag repeats back onto the chromosome ends. It counteracts the shortening from the end replication problem.
So it rebuilds the aglets.
Perfect analogy. It keeps the telomeres long enough, maybe not super long, but stable enough to allow continued division.
Where is telomerase normally active? Not in most of our cells, right?
Right. It's highly active in our germ cells, the ones that make sperm and eggs, and in some stem cell populations. Makes sense, they need to divide a lot. But in most of our regular somatic cells, after we're born, it's usually shut down or at very low levels.
But cancers turn it back on.
The vast majority, yes. They reactivate TERT expression, get telomerase working again, stabilize their telomeres, and achieve immortality.
Is that the only way? You said 85, 90%. What about the rest?
Good point. There's a smaller group, maybe 10–15% of cancers, that use a different mechanism called ALT, alternative lengthening of telomeres.
ALT.
Yeah, it doesn't involve telomerase. It seems to use mechanisms based on homologous recombination, basically swapping DNA strands to use existing telomere sequences as a template to lengthen others. You see ALT more often in certain cancers, like some sarcomas, glioblastomas.
Interesting. So telomerase reactivation is the main route to immortality, but there's this backup ALT pathway too.
Correct. But the take-home message is that overcoming that telomere-based clock is crucial for most cancers. Reactivating telomerase is the most common strategy.
Makes sense. And this isn't just cool biology, right? It has practical implications.
Definitely.
Right.
Let's maybe solidify some clinical pearls from all this.
Good idea. Pearl 1.
I'd say the Hallmarks of Cancer framework is just invaluable. It gives you a mental map for understanding the core biological hurdles cancer has to overcome.
Pearl 2. Telomere shortening is that mitotic clock in normal cells. The end replication problem drives it, leading eventually to senescence.
And Pearl 3. Telomerase reactivation is the key trick most cancers use to become immortal, to keep dividing indefinitely by maintaining their telomeres.
Okay, so how does this knowledge actually shape clinical decision-making beyond just passing exams?
Oh, massively. Understanding the hallmarks directly led to many targeted therapies. We target specific hallmarks.
Like EGFR inhibitors for sustained proliferation.
Exactly. Or anti-VEGF drugs like bevacizumab hitting angiogenesis. Even PRP inhibitors. They exploit the genome instability hallmark in cancers that already have trouble repairing DNA, like BRCA mutated ones.
Right, so targeting these specific abilities. What about telomerase itself? Seems like an obvious target.
It absolutely is, and it has been for a long time. The idea is attractive. Hit something active in cancer, but quiet in most normal cells. Telomerase inhibitors are in development, though it's proven challenging. The hope is to let cancer cell telomeres shorten again until they crash.
And how does radiation fit into this? Does it target specific hallmarks?
It hits several, actually. The biggest is inducing DNA damage, which relates directly to genome instability and can trigger cell death, counteracting resistance to apoptosis. Radiation also has complex effects on the tumor microenvironment. It can affect angiogenesis, inflammation, sometimes inhibiting them, sometimes paradoxically, maybe promoting certain aspects short-term. It's complicated, but definitely interacts with these hallmark pathways.
Okay, that really ties it together. Ready for a quick board blitz?
Let's do it. Hit me.
Question 1. A cancer cell stimulating new blood vessel formation to support its growth is an example of which hallmark? A. Evading growth suppressors. B. Enabling replicative immortality. C. Inducing angiogenesis. Or D. Activating invasion and metastasis.
That's got to be C. Inducing angiogenesis, building blood vessels.
Correct. Rationale. Angiogenesis is new blood vessel formation. Question 2. Why do telomeres shorten in normal somatic cells with each division? A. Increased telomerase activity. B. The end replication problem. C. Activation of senescence. Or D. DNA mutations in telomeres.
That's B, the end replication problem. DNA polymerase can't copy the very ends.
Correct. Rationale. The polymerase can't finish the job on linear chromosomes. Question 3. Which enzyme gets reactivated in most cancers to maintain telomeres and achieve immortality? A. DNA polymerase. B. P53. C. caspase 3. Or D. telomerase.
That's D telomerase, the key enzyme for rebuilding telomeres in cancer.
Correct. Rationale. Telomerase adds the repeats back. Final one. A cell population stops dividing after 50-odd doublings and shows high SA beta-gal activity. What state is this? A, apoptosis, B, necrosis, C, replicative senescence, or D, crisis?
That sounds exactly like C. replicative senescence, the stable growth arrest due to short telomeres, often with that SA beta-gal marker.
Perfect. Okay, before we wrap up, maybe a quick word on controversies or advances?
Yeah, well, one big challenge is that these hallmarks are all interconnected, right? You block one pathway, the cancer cell might just find a detour or upregulate another hallmark pathway. That leads to resistance.
Like whack-a-mole.
Kind of, yeah. So a big push now is towards multi-targeted therapies, trying to hit several hallmarks at once to prevent those escape routes.
Makes sense. Anything else?
Just that the tumor microenvironment is getting huge attention. We're realizing just how much the surrounding normal cells, immune cells, the matrix, how much they all influence the cancer's behavior in response to treatment. It's a whole ecosystem interacting with these hallmarks.
Right, it's not just the cancer cell in isolation. Okay, so let's recap the big points of this deep dive. We covered the original 6 hallmarks of cancer.
Sustaining proliferation, evading growth suppressors, resisting cell death, enabling immortality, inducing angiogenesis, and activating invasion metastasis.
Plus the key emerging ones, deregulating metabolism and avoiding immune destruction, and the enabling characteristics like genome instability and tumor-promoting inflammation.
We then zoomed in on telomeres, the end replication problem causing them to shorten, acting as that mitotic clock leading to senescence in normal cells.
And crucially, how telomerase reactivation in most cancers lets them overcome this limit, maintain their telomeres, and achieve replicative immortality.
And all of this is just fundamental for understanding cancer biology and how our treatments, especially radiation, actually work at a cellular level.
Couldn't agree more. Hopefully this has been a useful deep dive into some really core concepts for everyone listening.
Yeah, I hope so too. Thanks for joining us for this Rad Onc Smart Review RadBio edition.
Absolutely. Remember, you can head over to radonsmartlearn.com for practice oral boards, and please subscribe to Rad Onc Smart Review so you don't miss our next deep dive. Thanks so much for tuning in.